Thank you. It's been a pleasure to come and join the workshop for a couple of days. Very interesting, the mixture of talks that we've had. I'm going to talk of something different again. So my interests that I'm presenting today concern how swimming microorganisms interact with fluid environments. As I said, this isn't something we've heard about much yet, so I wanted to highlight some applications where this may be important. So what I'm showing up here, let's see if I can get this to work as well. No, we're going to use a stick. Oh, here we are, little green dot, I think. Maybe I'll use a stick, be more reliable. So here's a cartoon, lovely graphics that Roman Stocker's lab produced, showing a drop of ocean. Here we see bacteria tracking this lump of marine snow, which exudes nutrients, and this marine bacteria is doing chemotaxis towards it. We've got a copepod chasing around, emitting pheromones to attract its mates. So yes, we have a drop of very dynamic ocean here. Schematic of biofilms. As I'm sure many of you are familiar, biofilms occur in medical situations, also in environments, say in boats. So understanding how bacteria interact with their flow environment and form these films is interesting. Reproductive tracts. Sperm have to swim through this often non-Newtonian fluids along the way. So several examples of motile microorganisms moving in fluid environments. So what I'd like to talk about for the next half hour or so, I've got a taxi booked at 5, so I'll promise I won't overrun. I'm going to start by giving a sort of general overview regarding motility and patterns, both of which are in the title of the workshop, so I feel we're on topic. And a couple of particular examples I'd like to mention is gyrotaxis and bioconvection, which are very nice examples of where these swimming microorganisms can interact with the flow to generate spatial patterns. I particularly like to include those because whenever I'm talking to people about my swimming bugs, I try to convince them that they're brilliant swimmers because they can often swim at, say, tens of body lengths a second, which, you know, if you talk to a swimmer, that's pretty fast. None of you could swim at tens of body lengths a second. However, people will then say, but they're pretty small, so is it really important? And I think what's interesting is their interaction of swimming with the flows can actually generate some quite striking spatial patterns. So even though they're small and swim relatively slowly, they can still generate patterns. I'm then going to talk a little bit about population models, and I know there's people here interested in this idea of if you have individual level descriptions of motile behaviour, movement patterns, and we've had talks on this, how do you scale up? So how do you link individual level descriptors of swimming speeds, turning, and so forth to models which describe populations? So I will talk a little bit about that. And then for the sort of second half, I'll give a specific example of some recent research which I did looking at how bacteria can be trapped in regions of high shear that was motivated by some recent experimental data. Okay. So the first example I want to present about motility leading to spatial structure is something. This is a paper that came a few years ago out of Roman Stocker's lab, but had its foundations much earlier on in models of gyrotaxis that Tim Pedley and John Kessler did. So what we have here is a cartoon of a swimming algae. This is an example called Chlamydomonas, which swims in a kind of breaststroke way. It has these flagella up the top, and it swims. Now, these are algae, they're phytotaxis, they want to swim up towards the light. And one of the mechanisms they do that is by being bottom heavy. So in the absence of any flow, they're bottom heavy, they swim, and they tend to swim upwards. Okay. However, if you put them in a shear flow, as indicated by these blue arrows, you'll also have viscous torques trying to reorientate the cell as well. So if you have a viscous torque and you have a gravitational torque, what happens is the cells no longer swim straight upwards, but they'll swim at some angle to the vertical, as indicated here. And this angle is going to depend on the strength of this shear flow. So if you are in a reasonably low shear environment, cells are just tilted away from the vertical, so instead of swimming straight up, they swim at a slight angle. Likewise up here, where the shear is fairly slow. However, in this middle turbulent region where the shear, I say turbulent, I shouldn't say turbulent, strong shear region, in the middle here, the shear is so strong that the cells are rotated around and are trapped in this region. And what was particularly nice about this paper was that not only did they do the models of this, they were actually able to generate a trapped layer, a thin layer of this algal species Heterosigma acacia when they put these between two belts and generated this shear profile. So an example of where motility was leading to spatial structure, and not just in a lab, was why this was interesting, is because in the biological oceanography literature, people were very interested in this formation of thin layers of chlorophyll max, these phytoplankton layers, and they were associated with various ecological situations, high concentrations of sort of predator-prey interactions and so forth. So this was a possible mechanism, a new mechanism for generating these thin layers. Those are the hydrodynamic mechanisms as well, but I think this was a nice example of showing how swimming interacting with the flow leading to spatial structure and patterns. Another example I like to show, this is, it's lovely to come back and visit Cambridge, so I was here as an undergraduate, and I think bioconvection was probably one of the first things which got me into biological fluid dynamics. So this is a little movie of a phenomenon called bioconvection of some algal cells that I took. It's a dish of algae, about so far just a flask of algae, quite a dilute suspension of cells, and spontaneously we get these patterns forming in the cells. I say this is if you grab a cell culture, we're not thinking of gels, this is just a liquid culture, so 10 to the 6 cells per mil is about the concentration, so they're literally swimming around in a fluid, and spontaneously we can get these patterns forming in a manner of minutes. And what's happening here is swimming interacting with flow to generate patterns. So here's a sort of schematic of what's going on here. The cells tend to swim upwards because they're phototactic. Because the cells are denser than the fluid, you get an instability like Rayleigh-Bernard convection. That instability generates flow, and the flow causes the cell to focus into the downwelling regions because of this viscous torque. So again, here's just a little example of how swimming interacts with flow to lead to spatial structure. And there's lots of nice mathematics to do with that, which I won't discuss in detail today. So the mathematics which I do want to discuss is associated with how do we link what's happening to individuals to population level descriptions. So we have our little swimmer here, which I discussed before, and we might have individual level understanding of what this swimmer is doing. It may have a certain swimming speed. It may be affected by the fluid in the sense that it's advected by the fluid. It may be rotated by the fluid. And there may be other mechanisms which you want to include at the level of the individual. Later on I'll talk about bacteria doing chemotaxis, and there's lots of well-established biological models of this sort of run and tumble and other mechanisms for reorientation mechanisms. So we want to capture this behaviour. Clearly we could capture it by doing individual-based simulations, and there's space and usefulness of doing that type of thing. However, I'm going to talk more about continuum models, and I think these can offer some interesting and useful insight into physical mechanisms. Also, it can be computationally beneficial to have continuum models as in addition to individual-based models, certainly when you want very large numbers of populations. So, I've managed to. I think I did some animations, but left. This was supposed to appear in a moment, but we'll make it all appear now. So let me describe my sort of basic equation that I'll be working with for most of the talk now. So what I will be keeping track of is the probability or the density. Mathematically, I'm happy to switch between the two. Of a cell having a certain position at a certain time, but also keeping track of what its orientation is, its swimming direction. So this is key, keeping track of the swimming direction is key if we want to specify how it reorientates and what the effects of things like fluid rotation are. So the governing equation is simply a conservation equation. I'm assuming that in this model anyway that we're looking on timescale short compared to cell death, cell growth. And so this probability changes with time because of spatial kind of translation and changes in orientation. So changes in space, what moves the bacteria? So x dot represents advection by the fluid. Particles will be moved by background fluid flow. So U is the background fluid flow. Particles will move because they swim. So P is their swimming direction, Vs is their swimming speed. This term, I must admit, I don't like the look of it there. This is the convention that is in this lovely review paper by Santon and Shelley. If you plug it in, that's just diffusion, okay? It comes out the wash, but yeah, that's just the regular spatial diffusion, translational Brownian diffusion. This term here is looking at how do cells reorientate. What's the rate of change of cell orientation? And this comes from the fluid dynamics. So these two terms here come about by looking at what happens at low Reynolds number fluids. So we're thinking of very small particles. When we put small particles in flows, you have force balances, you have torque balances, and by looking at the torques on rotating spheres or rotating ellipsoids, you can determine that what the angular velocity of this spheroid is, and therefore determine what the rate of change of orientation is. And there's two components that are identified here. So the first one, if we set beta to be 0, that would represent a sphere, and the effect of the fluid on the sphere is to cause it to rotate at half the vorticity of the fluid. So this is representing the effect of rotation by the fluid. Now, later on in the talk, I'll be talking about slender bacteria. The other part of the effect of flow, shearing motion on objects comes out through here. Beta is the shape parameter, beta is 0 for a sphere, it goes towards 1 as you get to a rod, and this is the fact that cells tend to align with the principal rates of strain of the fluid. So if you have straining motion, rods will tend to align with that. So these are classical results from Stokes come from Stokes equations, Stokes flow equations. The other reorientation terms, this represents for gravitactic cells. This represents the fact that it tends to align with the vector k, the vertical. And then the final term is, again, written, I find, in this rather slightly strange, well, strange to me expression, but this is just rotational diffusion. Now this rotational diffusion, it may represent Brownian rotational diffusion, although typically that's actually very small for these swimmers. So I think of this more as the kind of internal randomness that a cell has in its way that it reorientates, and that can be captured by that term there.
Your P is living on the unit sphere.
Yes, yes, I should have stated that. Yeah, P is on the unit sphere. So what I have done quite a bit of, although I'm not going to dwell on it too much today, is looking at how you can connect this type of equation to this type of equation where we've lost P. Okay, so how do we average out the P dependence? And in the absence of flow, when you don't have advection in there, it's quite a standard problem in random walks to go from random walks or biased random walks to diffusion equations or drift diffusion equations. It's a standard textbook thing to do. When you have flow in there, you can obtain drift diffusion or advection diffusion equations. So N here is now the concentration of cells, and N has advection by the flow U. It has a mean swimming term, so Vs is the swimming speed, Q is the mean swimming direction, and it has a diffusion tensor. However, these mean swimming and diffusion tensor depend on the local shear. Furthermore, so here's some references to some work that was done on that. However, it only sometimes satisfies this advection diffusion equation. If straining component is too strong, it doesn't. The reason I put this lovely picture on, this is some work that John Taylor did, looking at bacteria. So this is, which way round is it? I think C is the chemical concentration, and this is the bacteria concentration. And this was some nice work looking at how bacteria undergo chemotaxis in turbulent flows. And the reason I wanted to put this is to just remind me that this type of model has been used a lot and has been very useful to explore things like this. How do marine bacteria undergo chemotaxis in fluid environments? And there's utility in this. But as we'll see for the final example of my talk, this is definitely sometimes. And at other times, you really need to go back to this full equation in order to capture phenomena. Okay. When it does work, so here's an example back that sort of came in the tail end of my PhD of when it did work. An example where you have homogeneous shear flow. A very simple. This is a mathematician's biofilm. We have biofilm close to the surface is linear shear. You have chemical gradients that you move towards. In the absence of flow, just solving the advection diffusion equation with perfectly absorbing boundaries. Cells move and absorb, certain proportion attach for certain parameters which I can't remember. And when we ranked up the flow to some arbitrary example level for the example of this, you can really reduce the amounts that attach. And the reason for this is what's happening is the shear is rotating the cells around, so that instead of doing a kind of random walk, they do a kind of their trajectories. Each individual trajectory is rotated, so they diffuse and chemotax is much reduced, which causes much less to attach. This is just some, as I say, very simple cartoon simulations, shall we say, not to be compared with experiments. Another example of using this population level model. This was done somewhat more rigorously, including comparison with individual-based simulations. This is going back to the gyrotactic trapping in thin layers. So this was some numerical simulations where we had a flow profile. So this is the horizontal component of velocity, the red line. And what you see is that I've imposed a velocity profile with a sharp shear region in the center, the slope here. And that translates to a vertical swimming component given by the blue line. So what we have here is there's a vertical swimming component. They happily swim upwards all this region. The vertical swimming component drops right down when the shear goes up because they're being spun around, and then regains its value here. And also the vertical diffusion starts at this constant value, and when the shear increases, the diffusion coefficient also drops right down at that thin layer. And because of this reduction in both vertical diffusion and vertical swimming, you see trapping. So if we start with a uniform distribution of cells, the cells in this bottom region swim upwards, and they have this transient trapping phenomena where they're not swimming through this region, they're sort of hanging out there until eventually they escape and then move up to the upper layers. So some examples of using those advection diffusion models to look at how swimming interacts with the flow and can generate spatial patterns. Okay, I'm trying to remember what I've said now. Yes, I've got one more example before I go on to the bacterial chemotaxis. So one of the applications that I was quite interested in looking at in terms of this spatial dynamics of swimmers was in the context of bioreactor design. So these algal species, people were interested in growing them up commercially, possibly not for biofuels, but maybe more for the high value sort of lipids in the sort of food supplement industry, and interested in trying to work out what might be optimal environments for growing these. So here's a couple of diagrams that came out of work that Martin Beasley Crows did. These are some sort of example bioreactor systems. And this is an example showing how we have to be careful when it comes to looking at swimming particles and not treating them as passive tracers. So this is a diagram that's representing some experiments that John Kessler did back in the 80s, which is showing these gyrotactic swimmers. So here's a little swimmer. Remember, it's doing its breaststroke, and the shear profile is such that because the flow is faster in the center of the tube, this is just a Poisson flow going down a tube, this high shear in the middle compared to low shear on the outside, cells tilt over and focus towards the center of the tube. So you get this phenomenon of gyrotactic focusing. Regular passive tracers don't do this. Okay, so if you were doing a chemical engineering problem and wanting to know where's my tracer, you'd get very different predictions than these active swimmers because of this focusing. And we looked a bit at using some of the mathematical theory to quantify this in some more detail. So if we had a pipe with a blob of cells in the center, put in Poisson flow, what happens to those cells? And we compared active swimmers, so these are gyrotactic swimmers, compared to a passive solute, which in the absence of flow would have the same properties as our active swimmers, so would diffuse with the same macroscopic diffusion. And what we see is that over time, the active swimmers, they're moving down with the flow, but they tend to move towards the center of the chamber, indicated by the left-hand line. So this is a half slice of the channel. Whereas the passive solute tends to disperse radially. And if we look at the drift along the axis and the effective diffusion along the axis. So this is very much comparing it to classic Taylor dispersion ideas of the effective dispersion of a chemical along a pipe. What we see is an order of magnitude difference. These numbers are a bit small, sorry, but with the effective dispersion, there's an order of magnitude difference between these gyrotectic active swimmers and this passive solute, and their along axis drift is increasing rather than going to this moving with the mean. And this is basically because the cells are all moving to the centre of the channel where the flow is fast. So they're obviously moving much quicker because they're going with the central speed rather than the mean drift. And they're dispersing much less because they're not sampling all across the channel. And hopefully there'll be experimental paper to complement this very soon. Right, final part of the talk that I'd like to speak about is motivated by some fairly recent work that again came out of Roman Stocker's lab at MIT. So this is looking at bacterial chemotaxis in fluid shear. So here we have this microfluidic device, so this tube setup where they have basically quasi-flow going through the tube. And what they observed is, first of all, I'll show the results in the absence of any chemical cues. So this is no chemotaxis, this is just the bacteria. What they found is that initially, in the absence of any shear, the bacteria, these little black dots, are uniformly distributed across the channel in the y direction. And the black line is the distribution of bacteria. When they turn the shear on, what they find is that the bacteria are depleted from the centre. So the green line, then the blue and the red are all showing how the concentration is depleted, is reduced at the centre, y equals 0. Okay, and then this was just to show that it's a motility effect, so this is the result with dead cells. And what was kind of interesting, well, what struck me as very interesting about this is that if cells satisfy a diffusion equation, no drift because there's no chemotaxis, so if they satisfy just a pure diffusion equation, even if you had some very fancy expression for the diffusion, you would always get a uniform distribution at steady state. Even with the presence of shear in there, it wouldn't be sufficient to generate this non-uniform distribution. So I was kind of curious as to whether going back to the full population level model where we keep track of orientation could be used to explain this model, and it turned out that it could, so that was very exciting. So I'll talk you through what we did. So, as I said, the spatial distribution that was observed can't be explained. Assuming it's the equilibrium steady state distribution, it can't be explained by a simple advection diffusion model. So what I wanted to do was return to this probability distribution function psi. So previously I went through in detail those terms, and then so this was to do with advection by the fluid and spatial swimming. This was to do with reorientation by the flow. And because I wanted to look at the effect of chemotaxis, I also introduced these extra terms, and these represent the run and tumble like behaviour of bacteria. So what we have is lambda is the probability of tumbling, of changing direction, and k is the turning kernel. So this represents cells which tumble that were swimming in direction p and tumble to some new direction. This represents cells which were swimming in some other direction p primed with some probability, go from P prime to P, and then contribute to psi. And the simplest way of introducing chemotaxis into this type of model is by saying that the turning rate, lambda, which depends on what swimming direction you're in, depends on P, is given by this expression here. So it depends on your direction relative to the gradient in chemical. So grad C is the chemical gradient. So when you're swimming towards your food, smells good, so you don't turn, or you reduce your probability of turning. So this is just a simple chemotactic model. So we plug this in, simplified things. So in the first instance, I thought, well, let's constrain things just to a 2D plane. Now, obviously, the cells in reality swim in 3D, but just for the mathematical simplicity, I'm going to see how good can we get if we just do a 2D constraint problem. So in that case, that governing equation reduces to this, where I've non-dimensionalized various things. I've non-dimensionalized on the channel width and on one of the diffusion timescales. So what do we have? We have Poisson flow. I'm going to assume isotropic tumbles for simplicity. Then the key parameters in the problem are, we have PE, which is the Peclet number, so that represents how strong the flow is. And we have a parameter epsilon, which is usually small, and that is kind of the swimming Peclet number. So that represents the swimming speed relative to the, it represents how far cells typically move relative to the channel width before they reorientate due to rotational diffusion. Little d is the ratio of spatial diffusion d and diffusion due to the cells swimming with speed s and having this rotational diffusion dr. Typically, this d parameter is quite small for swimmers for the bacteria. And I will be doing some asymptotic setting d equal to 0 in a slide or two. Chi's chemotactic strength. Sigma is just a parameter representing the ratio of the two different reorientating mechanisms, the tumbling and the rotational diffusion. Now, I'm going to run for it at 5, and I'm feeling sad that I'm doing that, because I think there's some of you who'd be experts and be able to help me with this, but we'll maybe you can email me afterwards. I'm a mathematician, and whenever I'm teaching, I'm always very insistent, well, what are your boundary conditions? And I think I feel like I've fudged the boundary conditions a little bit in this problem. So here are the boundary conditions that I looked at. So this is the cell concentration n, the integral of psi over p. If we took the governing equation and integrated it over cell orientations p, we'd have this expression here, where I view J as a cell flux, because this is a standard cell conservation equation. J is cell flux. So my instinctive boundary condition is to say I want to impose 0 cell flux at the top and bottom walls. I don't want cells escaping through the top and bottom. So that means I want to take the component of J perpendicular to the wall to be 0, which gives me this constraint. So the integral of this quantity at the walls must be 0. And that, to me, is in the moment, it's natural boundary condition. But when I worked with my collaborator for the numerics, we realised that this wasn't sufficient. It wasn't a sufficient boundary condition to ensure a unique solution. So what we did is instead impose pointwise this condition that this was equal to 0. However, in the asymptotics, I'm going to actually impose this, because I'm going to look at a simpler system with d being 0. So I'm being honest and open, and I feel there is a little bit of a fudge there. Anyway, here's our results. So what we have is the stars are experiments. The solid line are numerical, blue and black are numerical simulations, stars are experiments, and red is some asymptotic results. What we have here is increasing the flow strength, the Peclet number. There's no parameter fitting in this, in the sense that all the parameters used for things like swimming speed, reorientation, cell aspect ratio were all taken from the experimental paper. So given that, I was quite pleased with the agreement. Clearly we see reduction in the cell concentration at the centre, and therefore accumulation at the boundaries. I think that the most questionable thing really is what's happening at the boundaries, and that is problematic, and I think that needs resolving as the biggest thing to understand the experiments and the numerics. But what I'd like to just talk through now is the asymptotics, because, well, actually I won't talk. First of all, I'll say about how it affects chemotaxis. So there were some experiments, the blacks, when you had a chemical gradient. In this case, there's one parameter that's fitted. So in the no flow case, we fit the chemotactic strength, and then the others were no more fits for that. So what we see is that, again, the flow modifies the chemotaxis. It pulls down the concentration at y equals 0 at the centre of the channel. So I have got a couple of minutes. So I feel slightly cautious about showing my asymptotics. There were some beautiful talks this morning about showing wonderful convergence, which was very nice. So my asymptotics, let me think, my purpose of my asymptotics are to gain some physical insight as to why do we see this depletion in low shear and aggregation in high shear, okay? So I make no claims that the asymptotics are helping understand convergence, but they do help me understand the physical mechanism of what's going on. So with that caveat, let me talk you through my asymptotics. So I'm going to consider the case where D is 0. So this is saying that we're neglecting the spatial diffusion. There is diffusion in the problem, but it's all generated through the cell swimming, no kind of Brownian diffusion. Why did I consider this? Well, this is one of these where the story is going back to front. When I first did the asymptotics, I never worried about spatial diffusion. It was only when we did the numerics I realised this is going to be really hard without the spatial diffusion. So we're looking at no spatial diffusion. I'm going to take epsilon, which is the swimming Peclet number, to be a small parameter, which is reasonable for the given parameters. And I'm going to take psi to be given by this asymptotic approximation. When I substitute that into the governing equation for psi, F0 turns out to be the leading order equilibrium orientation distribution. So these terms, well, this term represents the effect of flow on the cell orientation. So this is just the rotation at the local, the half the local vorticity. This is the beta, the alignment with straining. This is the rotational diffusion. This is less important. This is the sort of the reorientation due to tumbling. So that's what F is. And the parameter N, which is the cell concentration at leading order, well, the leading order solution for the cell concentration, turns out to be proportional to this exponential function of chemotactic strength. That's expected. That's it's standard chemotaxis models. But it's also inversely proportional to this quantity VMS, which can be interpreted as the mean squared vertical swimming speed. So F is the cell orientation, and sine theta is the y component of swimming. So the mean squared vertical swimming speed is inversely related to ny. So, saying that again, the concentration is inversely proportional to the mean squared vertical swimming speed. So if we look at the orientation distribution, as a function of theta. The lines represent increasing flow. So as we increase the flow, so as we move towards high shear, what we find is there's peaks in the orientation corresponding to theta being pi and 2 pi. So that corresponds to the streamwise x direction. So the reorientating mechanism generates peaks in the orientation in the streamwise direction at high shear. So if you've got a peak in the streamwise direction, you see a reduction in the cross-channel swimming. So peaks in the horizontal means you get this drop in the mean squared vertical swimming speed. So you have reduction in VMS, and you'll get an accumulation of n of y. So this corresponds to what we see at the wall regions where it's high shear. We have enhanced cell concentration, and the centre line where it's low shear would see a reduction in ny. Okay. So just to conclude, a few comments. I very much enjoy studying swim microorganisms, and I think they're important in a lot of different applications and aren't just passive drifters. So questions that I've sort of tried to address a little bit is looking at what spatial distribution is, and how's transport affected by the interaction of swimming and flow, because they're not just passive tracers. And I also think this question of active suspensions, there's lots of mathematics. So asking what are the right continuum models, I think is important mathematical questions. And I've not even mentioned any like cell-cell interactions, or in the case of these chemotactic bacterium channels, I've completely neglected the whole interesting research which goes on about how cells interact with boundaries. So there's lots of nice math there. And just to mention, these are the various people who I've collaborated on the projects that I've spoken about today. Thank you.
Depending on the taxi driver, we have time for a couple of questions. Is there anyone? Sure.
Great talk, Rachel. This issue of the boundary condition, I'm wondering, you've used the equation for n, which is integrated out the orientation information.
Yep.
But in the presence of a boundary, you might expect the alignment vector p to actually become more and more orientated with the boundary through the hydrodynamic interactions. Do you think that maybe going back to that master equation for psi and modifying the Jeffrey equation for p dot to allow some sort of bias might fix up some of the oppositeness?
My quick answer would say possibly. I think looking in detail about what's happening at the boundary is going to be really important. I mean, the thing is, you've also got all your flagella. So if they're pullers compared to if they're pushers, and how that interacts. So yes, I think it needs studying, but I don't know whether I can simply say yes, that would be the way to do it. Yeah.
I think if I look at your boundary condition, right? So you had something like a sine of theta times
yes.
psi
Yep.
minus some positive quantity, I think, times deep psi dn, or d.
This one here.
Yeah, the
Yep.
one over here. Yeah.
Yep.
So could it be that if the sine, of the sine of theta, if that one is positive, that you're losing positive definiteness of the operator.
Of which operator? Sorry.
Of the if your diffusion thing. Because if you're
Well, I'm not. Thing is, I'm not assuming. So in this model,
yeah.
I'm not assuming that we can move to a diffusion-like operator. This is so. I'm making the claim that I don't think you can switch to a diffusion-like model.
Right.
So it's working out what would be these boundary conditions for that situation. So I don't. Yeah, because certainly this issue with losing positive definiteness can be a problem as well. But I think this is worse than that. That you can't do diffusion at all. Yeah.
So C is probability. Is it made sure in the second version of the model, the bigger model that you introduced, I think 2016 was there?
This one here?
No, later or even before.
Sorry, this
Right, exactly.
expression here.
Is it made sure that C remains bounded from above as probability?
Psi is bounded from above. So the integral of psi over x and p you could fix to be 1. And it's a conservation equation, so
So it's probability in the global sense, not locally.
yeah. I mean, I sort of, I'm a bit loose with switching between cell concentration and probabilities, but yeah, depending on how you normalise it.
The next thing.
Yeah.
So it's 5 o'clock. I think we should start there. Both speakers. Fantastic.